of people with, I guess, electric things coming from everywhere in the world. So a large part of the electric panel was down. So no more electricity in Octopus and no more Wi-Fi in this part of the building. But do not tell the guys from the electricity that I had a look myself or I'm fired. But it should work now. So as I am multitasking, so in this session 3, we are going to, we are in room 6, Kraken. So Nina, you're here, of course, Lara, yes, and Gabriel. Okay. So, I don't know if we are good or not.
Yeah, it looks like it. I will check, but it looks good.
Okay. Octopus is good too? Also?
Yeah, my WhatsApp is exploding probably because everybody has it. Yeah, all good.
It's very funny to have 6 presentations at the same time.
Looks like it.
So, at the same time in your jury, you will have Pelle Arberg from University of Gothenburg, Nadine Amiel from University Côte d'Azur, and myself, Christophe Motti, also from University Côte d'Azur, and also a bit from. So, Nina, since you're ready, so I will not just introduce your presentation. I think you're much better fit than me to introduce your presentation. Do not forget you have 15 minutes, and I will be very sharp with the 15 minutes. And then you have 10-15 minutes of questions. And also, if your supervisor, because now we are live, yeah, so if your supervisor also wants to ask a question on the YouTube live, they can. Sound good? Here we go, you have the floor.
Set some stuff.
Thank you.
So good afternoon, everyone. Thank you for coming to see my presentation. My name is Nina, and I'll present to you today the results of my thesis about the demography of the whale seals and their changing fast ice condition in Antarctica since 2006 in Netherlands. For a bit of context, as you may know, the Earth is facing global change, and especially at the poles and for ocean systems. And the sea ice ecosystems are in the front line, especially, for example, in Antarctica, with different areas over the continent being impacted by different factors, and different parameters also being impacted, such as the sea ice extent, as you can see here, with the average for example, 2023 being way below the median for the past decades. The question in science is often how to assess these changes, how to draw the link, between the biotic and the abiotic parts of the ecosystems. And one answer to these questions is to use sentinel species, and especially marine top predators, since they gather together different steps of the whole cycles of the ecosystems. And in this objective, the Sentinels of the Sea Ice, so the Sensei project, which I was part of during the thesis, proposed to use both seabirds and marine mammals at the both poles. And in Antarctica, we will focus on the whale seal. The whale seal has been studied on different aspects, such as diet, behavior, physiology, movement, quite well known, but about demography and population dynamics, all the work that has been done has only focused on the Ross Sea by the American teams, but not much elsewhere in the continent. So that was the scope of my thesis, to do the same, but in Adélie Land this time, on the archipelago of Point Géologie. So a bit more about the whale seals itself. It's an ice obligate species, meaning that they relate on ice to give birth, to nurse, but also to rest and to avoid predators. There is important difference in terms of life cycle between the both sexes, with the females being on the ice with the pups during the breeding seasons, while the males are mostly underwater, depending on territories. And it's a species that is monitored all over the continent thanks to mark recaptures protocols. So as I said, they are they're ice related and ice dependent, and they rely on a particular type of ice, which is called the fast ice, which is stationary type of ice, grounding to the continent or the seafloor, as you can see on figure 3, the seafloor being the dark blue. And this type of ice exhibits spatial and temporal variation all over the continents. We are, for example, here, that's Point Géologie, where my data were taken, and this is McMurdo Sound, where the American teams work. This figure shows the average timing for maximum of spice ice according to the months of the year. And for example, in Point Geologie, it's around July, while in McMurdo, it's way later in the year, and it's over like October, November. So we really have important differences in this topic. So with this context, my objective were first to verify, correct, and update the whole captured data sets that we had since 2006. And from this, to try to estimate the survival, temporary migration, abundances, and population growth rates for the adults in congeology at evidence. And at the end, to try to explore the link between the demographic parameters we estimated before and the sea ice dynamics. In terms of what we expected, according to literature for survival, for example, we're expecting something higher for females and for males, since males are fighting for the breeding territories, and it's shown that they have shorter life expectancy. Temporary migration, we were not really sure where we were going yet. We had a different point of view from the literatures, so it was a bit more exploratory. And for capture probability, we were expecting something higher for females than for males, because females are on the ice while the males are under the ice, and the protocol is conducted on the ice. In terms of the impact of the change in sea ice condition, we were expecting this to impact the different parameters and also to impact the feasibility of the protocol implementation. Concerning the methods, so quite a classic capture marker capture protocol conducted since 2006 using RFID tags that are controlled using a tag reader on the individuals. And this is the study area. So the white is the continent, the base camp is here, and this is the sea that it can be covered with ice depending on the time of the year. So we focused only on adults with a total approximately of 550 individuals with both females and males. And we also divided these individuals between tag groups, since change in type of tags due to detectability issues occurred in 2010. And from this, we were able to obtain the encounter histories with basically a line of 0 and 1, with the 1 being the individual has been observed and 0, it hasn't been observed. In terms of covariates for the sea ice, we choose the sea ice concentration that is expressed in percentage, and fast ice extent using the shortest distance from the Mongeauville station, so the base camp, to the open water as a proxy of the extent of fast ice. And we choose those variables for October and November, which is the period of breeding for the seals and also the period where the protocol of tagging is conducted. Then parameter estimation was conducted all with the software MARC with two different approaches. First, the Cormac-Joly-Seber, only accounting for survival and capture probability, and then the robust design, accounting for periods of times within the years, and allowing to account also for temporary migration, recapture probability, and also abundance estimations. Before going for the analysis, we checked, of course, with goodness of fit to the data using UCARE, and for the closeness of the population within the years. This is an assumption of the robust design approach, and we use close test for this. In terms of result, the first test of goodness of fit indicated a lack of fit with a bit of trap dependence, but a really important effect of transience. So the individuals coming in the population being seen once and never seen again, exiting the population. To correct this, we used what we call apparent age classes to distinguish the transients and the resident individuals. And from this, we were ready to go. The text was okay, and we conducted the analysis. For the closeness of the population, except few years where the population remained a bit open, the assumption was verified and we were ready to go. For the sea ice, this is on your left the sea ice concentration, and on your right the sea ice extent with the average values in orange, and we got also November and October. As you can see, there is like important variation between the years and even within the same years, like in 2021. With in blue you got November and this is October, so really different between the months. For the results of the model selection, the best model presented a survival changing according to the tag type and the transience or the resident status of the individuals. We had temporary migration divided in two types of gamma. So gamma second, the probability of exiting the area for the individuals, which was changing according to sex and tag types. For the probability, once the individual is outside of the area, to stay away the next year, gamma first, it was only changing according to the sex. And the capture probability was equal to the recapture probability changing according to time and sex. We also tried different covariate models with the CS extent and CS concentration, and the best one was with an effect of the CS extent on the temporary migration, the probability of exiting for the individuals. As I showed you, the best model suggested a survival changing according to the tag type, which doesn't have any biological significance. I mean, it's just different size tag for the cells. So this is the second best model with the sex. And we have a difference in transience and residence, with females having a higher survival than males in transience, while it's the opposite for residence individuals. But those results have to take carefully because all the confidence intervals are overlapping for both cases. In terms of temporary migration, the probability of exiting, we had something lower for the female tag before 2010, so with the first tag type. Again, higher spontaneity for the male tag before 2010. It's explained by the fact that we had only 10 individuals of this category, so it has to be taken carefully. In terms of covariate models, we had something quite clear with the temporary migration decreasing as the sea ice extent would increase, and with also like something way higher for the males than for the females, knowing that the gray one is again the first tag type males to take carefully. Probability of staying away of the study zone was lower for males than for females, but again, confidence interval overlapping. In terms of capture probability, we only had estimations since 2012. We didn't have enough data on the years before to have proper estimates. As we expected, we had something higher for the females than for the males due to the difference on life cycles, but again, overlapping confidence interval. And we also had, this is just for information, like the mean capture probability over the years, it's still increasing and it's quite good, especially for females where we almost reaching 1. In terms of abundance and trends, we ended up with a total of 82 females and 25 males. And as first, when we plotted the abundance over the years, we thought the population was experiencing a decrease. But when we looked at the confidence interval, it appeared to be stable. It encompassed 1, so we cannot conclude on any decrease of the population. And we also didn't find any significant effects on the CS extents on this. So for a bit of discussion, and to compare to what we expected, we did find a difference in females and males for survival, but nothing significant. Same for temporary migration. What we saw a bit more clearly was the capture probability difference. And we also had only effects of the CS dynamics on temporary migration. And as I will discuss later, we also had an effect on the protocol implementation feasibility. To put in a bit into context with what has been done by the American teams, they did find a difference in survival in females and males, but they accounted for age and they found different things according to the age groups. So it might be something interesting for us in the future to take into account. Same for temporary migration. They accounted for age and also for state. For example, if the individuals were breeders or pre-breeders, some females are also taking what we call a gap year between reproduction periods. So they accounted for all of this. And in the 2 cases, they had impact of the CS conditions on different parameters. And for the capture probability, they also found something higher for females. But again, they were accounting for more than we did. They took in account the age and the year, the birth cohort, for example. They also had kind of an impact of the CS condition on the protocol. But since I said before, the ice conditions are really different between the two locations. McMurdo Sounds, the American base is really more stable over the years and within the same year in terms of sea ice conditions than Adélie Land with archipelago we are studying. But they also had something stable for the abundance on the more recent estimations. So to talk about a bit the temporary migration that is affected by the sea ice extent, we have percentages that's quite high for the individuals once they're away to remain away of the area. And we have apparently, according to the models, a link with the fastest extent. As I'm going to show you, it's not really the detection that is involved, but the disponibility of the seals. And indeed an effect of the study zone, because as you can see, this is the continent here and here, and this is the ocean. These pictures were taken the same month, but a year apart. So as you can see, the sea ice condition can really change. In this case, 2023, easy, the seals are on the ice, they're available, it's easy for the field workers to find them. Well, in 2022, basically almost no seals were there pupping and breeding at this period, so nearly impossible for the teams to reach them or to detect them. So yeah, perspective to take into account the age groups and eventually also the states. The sea ice condition and the effect of the study zone to be able to compare more with other studies. Also the effort, because the condition for field workers there are a bit atypical. All the teams are changing each year, so depending which people you have in the team, they don't have the same effort. Some are really working harder than others as they can. And what's next? A bit something different, photo identification. The web access got fur pattern on the ventral sites, which allow us to identify them individually. And it's probably something I'm going to work on the coming months. And the goal is to try to make the same demographic estimation as we did, but only using photos, and in the long term, maybe replacing the RFID tags protocol by only pictures. So thank you everyone for your attention. If you got any questions, I'll be happy to.
Thank you, Nina. We can start with you, Albin, then Pere, and next presentation we will do the other way around.
Thanks for this nice presentation. So I would like to ask you, what about their prey? You are looking at the dynamic of the population of specific mammals. So, but what do you know about their prey and the dynamic of their prey?
Well, actually, that's a good question because it changed within, in the time of the protocol implementation. They're feeding on fish, like shallow water fish. And actually, I think I got extra slides for this. Yeah. Their diet is quite well known, it's quite stable, it's like the same species. And it's before 2010, it was quite all right. But in 2010, we had an event, which is the calving of the Mertz Glacier, which is like hundreds of kilometers on the east of the station, which is a really, I forgot the scale on the pictures, but it's really massive. And as you can see, it detached, and this part went on the west, and it completely changed the dynamics of the waters on the layers, the sea ice. And this is something we didn't have time to account for, but it might have also changed the stocks of the prey and also the dynamics and the foraging strategy. And also we only focused on summer, but we don't really know what they're doing in winter. There is already few research, but on the feeding strategies and stuff, but there is not much yet for this particular area.
Then, knowing about the dynamic of the prey, do you know if this can help you to anticipate the behavior and the dynamic of this marine mammal?
I mean, it's, as I know, it not has been done, but it would be interesting, yeah, to see the trends over the years and to link it with the sea ice, since a lot of species there are really linked to the sea ice.
Is there any village around?
No. Continence is not like it's inhabited in the sense of there is only scientific base or military base. So there is the I think if I go back to the map, there is the French base du Mont d'Urville, just there. So there is people all year round, but it's rotating. But there is nothing around. It's like
there is no
truly
magnetic pressure. No.
There is actually, but it's further away at sea. There is fishing a lot, but not directly on the coast. There is no activity except the scientific.
You hear the story with bycatch of this?
No, that I know for real. Like we had for other species, but they're not going that far offshore where the fishing boats are. So that we know nothing.
Yeah, interesting things. You said that you were just using the adults.
Yeah.
Yeah. Do you have any data on the pups and subadults?
Yeah, we do. Actually, when they're doing the tagging protocol, they tag all the individual they can reach, both sex and all the stages. It's just that the pups, there is a lot of pups that won't survive the first year.
Yeah.
And also for the wet seals, it's not really clear the definition between, for example, subadults and adults. So for us, for example, we counted that we considered adults all the seals that were more than 8 years old or that reproduced at least once.
Okay. Yeah.
But we do have data on the rest of the population.
Yes, because that would be really interesting to see.
Yeah.
See the survival rate of them as well.
That was
And maybe you also have the possibility to calculate the age of some of the individuals.
yeah.
If you know when they were
Yeah.
tagged the first time.
We actually got a lot of them that we know. It was more a matter of time this time because it was quite short. But it's the idea to do the same thing but for the whole population.
Yeah, because I think it's like you have a lot of more data that could be so interesting to explore when it comes to the total demography of the population.
No, for sure.
Yeah.
But also the database, I was the first one to a bit clean it and look into it this year, so it's quite fresh. We only had the clean database since I think March or April.
When it comes to the survival rate, they were like 0.9 or even lower.
Yeah.
Isn't that low?
It is a bit. We were a bit surprised actually by the numbers we got. We were expecting something much higher. And there's so many parameters that can enter the equation that, I mean, we're planning to work further on this, especially survival, like to look a bit.
Do you know the maximum age for the?
Well, we had, so it's higher for females than for males, and I think that the American teams recorded the females nearly 30 years old.
Yeah.
Something like this.
Sounds like a good estimate.
Yeah. So yeah.
Because then, for me, a survival rate of 0.9, and they can be 30 years old. It doesn't.
Yeah, that's what we thought too.
Yeah.
We're not like really matching with what we expected. So.
Yeah, because it's really interesting data, but I think you have so much more to explore. Actually.
Yeah, it's just the beginning of something really nice. I think. Yeah. Thanks.
Thank you.
Well, I may have missed some information, so I'm curious. I would like to know how you define closeness of a population and openness, I guess.
Yes. So it's. I got extra slides for this, I think, somewhere.
Well, you can try to go for it.
Yeah, it's just that in robust design we had two approaches for the models: primordial saber, which is one occasion per year, and robust design, which can be multiple sub-occasion within the same year. For us, we divided each year in robust design between 4 periods of 2 weeks, and one of the assumptions of the robust design, it's between those parts, the population is supposed to remain close, so there is no entry or no exit of individuals of the population.
But it's an assumption.
It's an assumption.
Okay, it's an assumption. Okay.
But you have, like usually in this type of studies with the robust design, you test it before to see if you're like really far from the assumption of the models or if you're quite close to it.
Something totally different, but looking at the patterns of the seals you talked about, do you know what creates this pattern? Is it a long-term DNA related something, or it's due to the environment and it can change with time?
That's a good question, actually, and we're actually looking into it because we don't really know exactly. It's more like pigmentation of the fur itself. It's not supposed to change over the lifetime of the seals, and we also
Supposed.
It's supposed, like it's not has been done for the species yet, so we don't really know. We also maybe gonna see if there is a link, for example, between the mother and the pups. These are like similar patterns or something. But it's really on the trial. Like we're still figuring out even like how does the software work. Like we have to adapt the software to the species since it's not has been done. But it's gonna be a nice investigation, I think. Yeah, I'm excited for this for sure.
So we were talking about the beginning of something that you have a lot of data to analyze. So you talked about age. That could be one parameter that could impact a lot.
Yeah.
What's the reality here? Is it a parameter you have and that you have also in your database and that you will be able to use or not?
We do have it for some individuals. For example, all the pups we tagged, we know exactly almost the day they were born, or at least the week and the year. So for this, we're gonna have, and since the database is building, we got more and more individuals. But for example, the oldest individuals.
The beginning of a cohort, you can start.
Yeah, exactly.
So there is no easy way to get the age of an adult, for example.
That we know, like for example, we thought about checking for the bloods or whatever, but of what I know, it not has been done. So it's like only observation like when they were born. But it could be also, yeah. I don't know, it might have been done on other seal species. I didn't have a look into it, but it could be also.
You can look at teeth, but then you do death.
Yeah.
Yeah.
Maybe.
Going back to you, Aldin.
Yeah, one more. So you started your presentation talking about global warming, climate change, sorry. So what can you conclude? I mean, what will you do with that?
Well, the objective is a bit to go a bit deeper in the part of CS dynamics, because we got a lot of data historically of CS dynamics on this area. It has been treated on different ways, but not on the link with marine mammals like this. So it would be a bit in this mindset that we would like to continue to focus on the species, since it's really relying on ice. And since the ice dynamics is changing due to temperature or melting, whatever, to continue on this.
And do you think that the dynamic of the biodiversity there can preserve the ice? I mean, can give you info to anticipate the melting of the ice?
Yeah, definitely. Well, actually, melting for Antarctica on the last research that has been done, Antarctica in a lot of areas tends to have more ice. For example, than the Arctic that is really melting and shrinking. But yeah, definitely that's the idea of the project I'm part of, Sensei, is really to use different indicators, like we got flying seabirds, diving seabirds, marine mammals, to study all of these indicators together and to see how we can anticipate and also implement this in conservation measures, political decision and everything. So, yeah.
Last question.
No, I'm fine, I think.
Okay. Do you have a question, guys?
Okay, we start.
Gabriel.
Is it age estimation based on blood samples?
Yeah, it's something that can be done on different species, on all the animals possible. I don't know if it has been done on seals, but it might be an option.
We'll talk about the exact same.
Okay. Okay.
No spoiler.
Stay tuned.
Okay, cool.
Thank you a lot, Nina.
Thank you.
And congratulations again. Here we go with you, Dilara. So for your presentation, just to make that clear, one member of the jury has been part of your through supervision. So this member of the jury will ask you questions, but will not participate to the grading, of course. Go ahead.
So hello everyone, I'm Dilara, and I'll be talking about my master's thesis that I conducted with Aldine Ariel and Eric Lassiner. So the team at IRCAM, the Institute of Research in Cancer and Aging at Nice, works on stress response regeneration and aging. And aging is important to study because aging and age-related diseases will be causing a heavy financial burden in the future for us. And aging itself is characterized by the loss of regeneration. So what is regeneration? It is a phenomenon wherein a stress response or an injury can lead to the launch of a developmental program wherein it restored the original shape and function of the lost parts. So you can see different species actually have regenerative capacity at different levels, but they lose it as they age, so they don't retain it throughout their lifetime, except a couple interesting and impressive species that have whole body regeneration, and they retain this capacity throughout their lives. So it's interesting to study them because if we can understand the mechanisms behind whole body regeneration, then we can apply it to humans as well. And interestingly, we know of one of these mechanisms, which is ROS, reactive oxygen species. And even though we know of ROS in a negative context, like the oxidative stress in mitochondria from the hallmarks of aging, which leads to apoptosis or DNA damage, we know that there are more neutral or even more positive biological processes that ROS signals towards. So some of these, whether they're positive or negative, do play a role in regeneration. So what is some research that has been done with ROS? We know that a couple of these regenerative animals has been studied in the context of reactive oxygen species, and they are zebrafish fin, planaria, and hydra. And as you can see here, there's an upregulation of ROS at the amputation site. So what these studies are missing are looking at the ROS metabolism genes. So that's where we come in. And here you can see a generic ROS metabolism gene pathway. And here coded in red is the ROS producing system, which produces superoxides, a type of ROS. And here the ROS converting system in yellow, which converts these into hydrogen peroxide, a different type of ROS. And finally, in green here, you can see the ROS detoxifying system with all the redoxins, etc. So how will I study these? I use Nematostella, and here you can see that it's in the sister group Bilateria, and it has a more ancestral position. It's an amphizoid. And for those of you who don't know Nematostella, I'll give a little bit of introduction in the morphology. It has a mostly clear morphology, tentacles, the oral opening, the pharynx here, followed by the thickened folds on the inside, which are the mesenteries, and along the body column at the bottom, there's the phylum. So why are we going for this model organism specifically is, of course, because of whole body regeneration, as we mentioned, because of its regenerative capacity throughout its lifestyle, and because of high ROS resistance from research that's been previously done in the lab that we know it goes up to 200 micromolars, which is very high, and because its genome has been sequenced and it has a very high similarity to humans as well. And of course, reproducibility, like all other model organisms, very culturable in the lab and produces a lot of eggs. So just to give you some information on the regeneration steps of Nematostella here on the earlier steps, which were important for the context of my study, here up to 4 hours post-amputation, we get whole body regeneration, and this is, wound healing. And then around 4 hours, we have the apoptotic stage starting. And this is wherein the mesenteries, as I mentioned, come in contact with the epithelium. And once this invagination occurs, around 12 hours post-amputation, we have the start of cell proliferation. And for the proliferation dependent stage, it's characterized in the beginning by the burst of cell proliferation. And the steps are the tentacles forming, the pharynx forming, and the end is when the oral opening forms. So, okay, it's downloading, I think something's moved, but. So for the genes, this was actually a study in the greater context. It was a PhD project in the lab done by Aurora William. And prior to my entrance to the lab, she actually looked at this Ross generic system, and she found 11 orthologous genes in humans as well that are in Nematostella, as you can see here with the black stars. So what is my research question? It is when and where are these genes associated with Ross metabolism expressed during regeneration? And my hypothesis is that the genes associated with Ross metabolism appear at the amputation site. So how did I tackle these questions? For the first part, I looked into the MDRT database. And this database has previous work from the lab that highlights the temporal expression dynamics of genes in Nematostella. And the 9 clusters that you can see here are the temporal gene expression dynamics of transcripts involved in regeneration specifically. And the 11 orthologous genes that I have are in R6 and R7. So a little bit of information about R6 and R7. Here you can see that they are biphasic. They have 2 peaks. And the first peak in both of them correspond to the apoptotic stage, as I mentioned earlier. And the difference is that for R6, the second peak is a bit earlier, around the beginning of cell proliferation. And the second one is a bit more spread out and later, around the burst of cell proliferation. So this was a more global view, and to give you a bit more insight with the producing and the converting systems. Here you can see the gene expression dynamics for all of my genes, and you can clearly see the peaks, especially with the converting system. The first ones corresponding to apoptosis, later on to the beginning of cell proliferation, and the ones that are in R7 appear a bit later and at the burst of cell proliferation. So for detoxifying, similar results, except the gene expression dynamics are slightly more muted and spread out at a later time point. So here, what can we take away from these results? It is that they are threefold. First of all, that they're biphasic, so 2 peaks. The first one corresponding to apoptosis, second to cell proliferation, all of them. And the third is that they appear in sequential order. So the peaks occur first for the producing system, and then the converting, and finally the detoxifying system. And for the second part, the main part of the study, which is the spatial expression, and how did I do this? Studying in situ hybridization, more specifically whole mount in situ hybridization that has several steps. And most important of those being hybridization, of course. And this works with the principle of nucleic acids annealing, complementary annealing, so using probes. And the several steps of antibody blocking, incubation, and development with our chromogenic stains, we can reveal a purple stain. So the interesting thing about whole mount in situ hybridization is that we can see where the mRNA transcripts are in the animal without having to dissect the animal. So from these images here with the purple stains, you can see for the Ross producing system, first, the mRNA transcripts are for uncut animals at the arboreal tips of the mesenteries. Following amputation at earlier time points, they are more at the amputation site and the arboreal tips of the mesenteries. Next, it's the entire length of the mesenteries, and at later time points, the staining disappears. So you can see similar results for the converting system, for which I did this similar experiment for all the genes. And here you can see the same results with the arboreal tips of the mesenteries, amputation site, entire length of mesenteries before residing. As for the detoxifying system, one interesting point was that at later time points, like 36 hours post-amputation, the staining still remains at the amputation site or the arboreal tips of the mesenteries. So looking at all of these staining patterns, I kind of proposed a model for the dynamic expression of these Ross metabolism genes in Nematostella. So here you can see the uncut animals have a staining at the arboreal tips of the mesenteries, then the amputation site in the arboreal tips, entire length of the mesenteries before going back to the arboreal tips and residing. And similar to our results from the WEN, from the temporal expression, you can see that the Ross reducing appears earlier, and then Ross converting, and Ross detoxifying finally. So what do these results indicate? First of all, that they are biphasic. So these two peaks that are very distinctly at 2 time points suggest that Ross might play a role in these two biological processes, cell proliferation and apoptosis, separately. And as we see the staining at the amputation site, we can say that Ross might also play an important role in regeneration. And thirdly, the staining at the abortal tips of the mesenteries corresponds to previous research from the lab, when they looked at stem cell genes in Nematocella, and the spatial patterning was in the same location, suggesting an importance of Ross metabolism genes and the correlation with Ross metabolism genes and these stem cell genes. So for my hypothesis, the genes associated with Ross metabolism are expressed with the amputation site. That's correct. But I also came up with some novel findings. Like the staining at the abortal tips of mesenteries are the potential stem cell locations in Nematocella. And the second interesting thing that was not in previous research was that the expression is biphasic. So there are two very specific peaks corresponding to two important biological processes in regeneration. So for further studies, there are a couple questions. It opens the door for more research. And first, does ROS inhibition affect regeneration? Does ROS inhibition affect cell proliferation and apoptosis? And thirdly, does ROS inhibition affect the genes associated to ROS metabolism? So how can this be done? It's using DPI, which is a pharmaceutical drug that blocks the NOX enzyme, so blocking the cascade up here. And there is also previous research in the lab. So these are some results from the lab where we know the answer to the first question. Yes, it does block regeneration at 100 nanomolars. And since I've submitted my transcript, I've been able to do a little bit of study with DPI as well. So I'll present my results to you. So here you can see with the DPI, you see the loss of a specific location, which is the abortal tips and mesenteries, and hopefully for people in the back, they can see that there is some staining on the epithelium, and staining is retained at the amputation site as well. So I will leave you with these very interesting results, without going too much into depth. And I'd like to thank my supervisors again and all of the students and PhDs in the lab for all of their help and support. And thank you. I can take your questions.
Judy Lara. So Pele, we have some pressure. Can
I must admit I'm not an expert in this field, but still really interesting to listen to. And I was thinking about, you did your amputation and you did it at the same spot always. Yes. Have you tried to do it in another site? See if this is something that could be important as well.
Regional team is not here.
Okay. Yes, there is research from the lab. Actually, there's also a paper in the bioarchive where, because it has whole body regeneration, we can actually cut anywhere and the animal is supposed to regenerate. And they did this research with different sections, and they found that when you cut the part of the phyla where no mesenteries are left, then the animal cannot regenerate, which is why we kind of know that the mesenteries are a potential location for stem cells. And also because there are some grafting experiments where they remove the mesenteries and the animal wouldn't regenerate it. When they're regrafted, they do. So yes.
Okay. But so why did you just use this specific site then?
For me, yeah. This was because so regeneration can either be with the cell proliferation or at the abortal tip here when you do regeneration, it can be without cell proliferation. So since this is kind of a study in the greater context, and there is also some research they're still doing with apoptosis, looking at apoptosis, looking at the rates of cell proliferation, etc. If I had done sectioning at the world tips, then they wouldn't be able to look at cell proliferation. So it was kind of to maintain the balance, and you know.
Yeah. So another question: if you put your results in a more general context, the big picture, what is this contribution to the field of aging, for example?
Yeah. So for aging, so as I mentioned, a lot of animals have regenerative capacity, but they don't retain it. And one of the hallmarks of aging is stem cell exhaustion, for example. So if we understand the whole body regeneration in these animals, and we understand how it comes to be, whether it's the stem cells or whether it's signaling with ROS, etc., then because we also looked at the genes and they're orthologous to humans, so theoretically we think that we can apply, for example, ROS signal to humans as well. And that way it would be regenerative therapy, but also there is some research that I won't go into depth with cancer and redox therapy. So taking the detoxifying system itself separately also would lead to some sort of therapy for cancer or aging.
Thank you.
Maybe coming back to basics. So talking about you were talking about the context. I will come back to apoptosis. Could you remind me how it works and what is the connection with ROS? What is apoptosis and what's the connection with ROS?
Well, to put it simply, it's like a cell death. Basically, it's a mechanism where the cell kills itself. So, what's this context with ROS, right? And that's your question. So basically, an accumulation of ROS is cytotoxic, and that kind of signals for the cell to launch a program within the cell to kill itself before it spreads, for example, like cancer, etc. So basically, maybe I have a thing for this. Basically, the role ROS plays. The role ROS plays depends on its concentration. So from more new research, we know that it's not just negative, not just apoptosis, but if it's at the levels where it corresponds to apoptosis, very high concentrations, then that's what it leads to. So that's its significance as a signaling molecule, that it can launch pathways like MAPK, etc., that will further down the line turn into apoptosis. So it really depends on the concentration of ROS.
So ROS is a species like a compound which is produced by the cell itself, or that comes from the environment?
No, by the cell itself. It can be from the environment, but it's the NOx enzyme that I talk about here, which is an NADPH oxidase. Here. NOx, and I was looking at NOx4 specifically. So this enzyme takes oxygen, it produces superoxide radicals, so O2 minus. So it's within the cells. Usually they're located at the membranes. Can be the oxide membrane, could be the mitochondrial membrane. So.
And on methods, maybe I missed it, but because sometimes I take notes and at the same time you say something, it's like when you're in class and you miss something. For the injection, I think this part was clear, but I would like to know for your first part, what was the part that you've done yourself? Were you doing experiment yourself, or you were using the database?
No, I was using the database. Okay. Aurora had the list of IDs, and I went and extracted the graphs, and I had to find time points to do the in situ that would give me some significant results. So that's why in the first part, I kind of look at the graphs and try to understand where I should start.
So this first part helps you to design?
Yeah, leads to design.
And also for the method part, the staining.
Yes.
How does it work? How do you stain? What exactly do you stain?
Basically, I have a probe, and then this probe has a deoxygenant molecule.
Yep, molecule.
As such, yes. And then I incubate it with an anti-dig antibody. So that has an enzyme, AP, alkaline phosphatase. And then I incubated further with FVTB-CYP, which is two chromogenic dyes. Have very long names. And
So what is colorful, actually?
Basically, this is kind of yellowish at first, and then turns purple because of the AP, basically.
Okay. So here you have wild types that you hybridize. No, but initially the organisms they are from a wild type. Yeah.
Yeah.
And could you imagine like also having some part of them that you could alter so that it could be colorful by itself? You will not have to do the hybridization.
Okay, maybe not exactly that way, but there are knock in, knockout experiments, basically, which is also the next step of it. So there's also previous research where they knock out SOD, for example, which is one of the genes I looked at, and that way you see how it affects regeneration.
Knockout, so it means you remove the gene.
Yeah, you remove it. So basically, that way you can kind of see how that affects regeneration, but then you still do it in situ. So,
okay.
Yeah, you come back to this part.
Well, do you have another question before we go to LD? No? No. Aldin, you're allowed to ask questions.
Question. It's tough.
I have more.
Okay. Yeah, I have one. Now that you know well your topic and you know the system, and so what you will do different in your project, and what you will love to do next.
So what I would do different with all the time and money that I have. So I would go back and do more replicates, and I would do more time points. With all the knowledge that I have now, I'd start with earlier time points, and those give most significant results.
Okay.
And yeah, I would do more replicates. What would I change? So yeah, I think those are the things that I would do differently. If you want me to go to some technical details, then I can also.
No.
Okay.
And what do you want to do next?
What I want to do next is the knock-in experiment. So that would be nice. Knockout, sorry, with the SODs and the knocks. And I would like to see how those.
Why those two pathways?
Because if they're at the very beginning, so I think they would have the most effect. So I would start from there, and if I have more interest in cancer, I kind of do, but first I'd start with that. And if I wanted to go more into that, I would do knockouts for the redox system, to see how it goes into the redox therapy and research into redox therapy.
Okay.
And also more into BPI. Yeah.
Thanks.
I think we're good on our side. So, Sam, one question from you as you're here. No?
Congrats.
Congrats. Allez, congrats. Congrats. So here we go now for Gabriel. Gabriel, you're not even sweating.
I even put on a shirt.
Yeah.
It's so cold. Now I brought it, so I have to use it. I just put the timer.
Yeah.
Not timer, stopwatch.
Here we go.
Hello everyone, good afternoon. I'm super excited to present my project today, and I think the previous two presentations are perfect to introduce this project as well. I was supported by my supervisors, Daphne Anastasiadi, Francesc Piferrer, and Maren Bernwater. I was lucky enough to work in the Barcelona Institute ICM and spent 4 months in the plant and food research in Nelson, in New Zealand. My project is titled Shared Methylation Regions, SMRs, a new method in comparative epigenetics to create a multi-species Piscean epigenetic clock. In order to understand what I did, I want to talk a little bit about genetics in general, epigenetics, which is considered looking at all heritable traits which affect cell function without altering the DNA information. And we're mainly talking about 3 processes, which are DNA methylation, histone modification, and microRNA regulation. And in my project, we will focus on DNA methylation, which usually occurs, it's a slight chemical modification, at the cytosine when there are cytosine phosphate-guanine pairs in a DNA strand. And we also need to know, and this is coming back to aging and everything we talked about before as well, that when we look at methylation, that we can see a trend in change, in methylation change, with the passage of time, or else the aging of the organism. So if we look at this schematic, for example, as the time passes, these are the methylation marks. Some are demethylated and others are methylated. With the change of time, the epigenetic marks, the methylation marks in this case, are changing as well. And in general, we're speaking about a global, so the whole genome, global demethylation or hypermethylation. And in promoter regions, for example, we're talking about hypermethylation or methylation of these regions. And these changes create patterns which we can use to estimate the age in various organisms. And this phenomenon is often referred to as epigenetic drift, so this pattern. And based on this phenomenon, based on this pattern, which occurs with age, in 2013, two research groups developed a statistical model which is called an epigenetic clock to estimate the age based on these epigenetic changes, the methylation marks, which estimates the age extremely accurate and very precisely. And this was first published in 2013. Then in 2020, there was another study published which developed the first epigenetic clock in fish, which was my two supervisors. And quite recently, in 2023, there was a universal epigenetic clock developed for mammals, which works on, or was done on over 180 species, which is supposedly to work for all mammals, going back to the seals. So now, with that knowledge, we came up with the question if we would be able to develop an epigenetic clock for fish, but not just one species, which has been done before a couple of times, but for multiple species. So multi-species epigenetic clock for fish. And basically my first attempt to work on this was to look at what data is available for all different kinds of fish epigenetic clocks. And for us available, there were about 13. Some of them are unpublished, as you can see here, and others are publicly available, including their data. And by selecting them through looking at is the data actually comparable within these, I basically concentrated these data samples down to 5 species. In the end, I had to reduce it to 4 due to a lack of comparable data, which were zebrafish, Australasian snapper, and Atlantic cod and European hake. These 4 candidates here. And they showcase, well, different characteristics. For example, they have very different lifespans, maximum lifespans, ranging from 5.5 years to 45 years. And in the data set, we're talking about data points I use to create an epigenetic clock. And these are here showcased as CPGs. Those were the cytosine points where methylation data was collected. And you can see there is a various amount of available CPGs within my data set, which makes the overall comparison quite difficult. Because the normal approach when you're trying to compare multiple species and try to create one model to estimate the age of all of them is that you want to have the exact same data points. This applies to other projects as well, but you want to have the exact same data points in order to compare them. In this case, I'm working with data originating from different authors, from different projects, from different experiments. And the result is that my data points indicated by this small circle, is not exactly the same among the species. So I have a problem, which is that I'm trying to compare data which is not comparable. Usually, and that's why I put it in brackets, usually when you're trying to compare multiple species, use methylation arrays to guarantee you have the exact same position. In this case, I haven't had that, so I had to come up with a method which has never been done before, which was quite exciting, to make uncomparable data comparable. I will talk through this quite briefly, open to questions, of course. So we're starting off by having single data points for all our species. I took these, which is positional data on the genome. I know where it is. I then expanded this position to a sequence. Why? Because I can't compare or align a single position on a genome, but I can align sequences. So I took my data points, expanded them to sequences, and then aligned these sequences for all the fish on one common reference genome. In my case, I took the human reference genome. Hope you're going to ask me why. And as you can see here, there are some regions on the common reference genome where you see the sequences for all the different fish are within a certain area. And this area I took and declared as shared methylation region. And this basically leads to the fact that I'm not comparing single data points with another, but I'm comparing regions where there are data points and then selecting one of them by correlation testing, but I won't go into detail, and then compare one data point within the region along the species. That was basically the solution for my initial problem. And with that, I'm able to create a statistical model which uses the methylation data you saw before and tries to estimate the age just on the methylation data. That's why you can use blood, for example, because there's epigenetic data in it. And briefly how it works, you have independent variables, which are your predictive variables, which is the methylation data, and you have age data. And then you're trying to train your model, your statistical model, on a training data set to try to optimize it and then test it or validate it with a validation or testing set, which is data basically that the model hasn't seen before. And by that you can assume and infer the performance of the model. And I did this with chronological age and relative age, and more on that later. Now we look at the chronological age of the species, and here you can see the output of the model. Here is the estimated age on this axis, and this is the observed age. And this line, this dashed line, indicates a correlation value of 1, which means that the estimated value is the observed value, which is what you want to get. And as you can see, this works extremely well with a correlation value of 0.9 and mean average error of below 1 year, which is already way better than most of the autoliv age estimation data, or with T, for example. And here you have the training set and the testing set, which I talked about before. And this basically means that since the values are quite similar and the errors are quite similar as well, statistically tested them, they're not significantly different, which is what I wanted to have. They're extremely accurate, work for all 4 species as well. And tried something else because this was not good enough. So I tested relative age as well. And when I talk about relative age, I'm talking about using the age of the fish and dividing it by its maximum lifespan. What it does is that you get something, not in years, but something, a value from 0 to 1. 1 meaning it's the age of the fish is at its expected maximum lifespan. 0 is at birth. And when you use relative age, you're reflecting different traits. We'll talk about this later, but what's interesting is that this worked extremely well as well, less well than for the chronological age, but you can see that these are nicely clustered along the line, which is what I wanted to have. Same thing goes for the testing set, which validates my results. And also these two mean average errors are not significantly different. Just on a clarification, when you have a zebrafish, which is 5 years old, for example, it's close to its maximum lifespan, so it's relatively speaking as old as the Australian Asian snapper, which is 50 years old. So chronological age, 50 years, 5 years, relative age, close to one. And it works as well. Now the differences I want to point out again, because it is quite phenomenal that this works, because the chronological age looks, they're different models, but they look at the same data set, which is methylation data. And within that data, there are biomarkers which reflect the passage of time. And this is independent of the species biology. It is one clock which ticks at the same ticking rate for all fish species I looked at. Whereas the relative age clock, as I explained before, since it's plotting all along the line, along the same line, the 3-year zebrafish is as old as the 30-year snapper and so on, has a different ticking rate. Otherwise, the 5-year zebrafish wouldn't be as old as the 50-year snapper, for example. And both models with the same data set are capturing these phenomena, which is remarkable. Now I also wanted to see if, let's say you want to include another species, which I didn't look at my project, wanted to use my same model on, what the performance would look like. So what I did was a LOSO analysis, a leave one species out analysis, meaning that in my training set, I have 3 species of my 4, and in the validation set, I have 1. Meaning that the model knows, okay, there are 3 species. Now I give it the data of 1 unknown species and see how it performs. And I did this for all the species, and I quickly want to go through it. This is the Atlantic cod, which works quite well. So this is one model, has never seen a cod before. It estimates the age of cod very well. Here's the hake, and this is something interesting because in ecology and in fishery sciences, we know that estimating the age of hake is extremely difficult using otolith data. And guess what we used here? It was otolith data. So the model is overestimating the age of hake, which could be explained by maybe the age data, underlying age data of the hake is very inaccurate. Here, something really interesting happens, which is that the trained model knows fish which are a maximum of 7 years old. But if I feed it data from a fish which goes beyond that, it also captures this quite nicely, although less accurate. But this could infer that this model could be used for fish which go beyond this range of age. And lastly, I want to point out this for zebrafish, which is a very short-lived species. The model, which has never seen a zebrafish before, looks at the zebrafish data and clusters it as a very short-lived species with a very young specimen, which, yeah, it's remarkable again. So quickly to summarize, we basically came up with a new method which could be used not only in aging research, looking at methylation data, but could be used as a general means to identify biomarkers, not only for aging, but basically everything you can correlation test. And with that method, we developed the first epigenetic clock, chronological clock and relative clock in fish for 4 different species. Thank you. Sorry, it was a little bit over.
Yeah, that's okay. Thank you, Gabriel. I don't know, Aideen, maybe you have a few questions about that.
Thanks a lot, Gabriel, for this wonderful presentation. It's a really, really dark topic, and it's actually, so if you have to help your model and to refine your data, so which kind of species can you add Which kind of fish species can you have here? Or you would like to have?
Yeah, because it's not a lot.
No, for species is not a lot. And we have basically a little bit of data for, or there are 13 epigenetic clocks for fish, which are available, or was available to us, but not all of them use a sequencing method where you have enough data available to compare it. So ideally, I would include a species with enough data to improve this model. I validated this as shown here. I validated this by leaving one species out, which is already crucial because I'm starting off with 4, and I'm only using 3. So I expect when I would introduce another species to optimize my model, that this performance for all would increase drastically.
But then if you think, okay, forget the data availability. So if you think at physiological level,
Yeah.
you'll be able to challenge your system, okay?
Yeah.
So which kind of fish species can you think including?
The best way to create a universal model or a multi-species model is to have fish with very different traits, with very different lifespans, with very different habitats, with developmental trajectories to get the bigger picture, basically.
Exactly.
And these fish are, relatively speaking, quite different. The cod and the hake are quite similar, but what would be even more important than maybe the selected species would be the accuracy of the data. Don't want to come back again to the data, but the age estimation, as you can see here, is extremely important as well, because you're basically feeding your model with that age data. So you want to make sure it's accurate. For this species, we don't know that it's super accurate. We know that it's quite inaccurate. Usually when you use autologous data, these were, this was in the lab, so you know the exact same age.
Do you know the killifish? The African turtle?
Yeah, so there's an epigenetic clock for.
Yeah, because it's 3 months life, and then in 1 week they got all the aging sign.
Yes.
So it's really it will be super crucial and interesting to look at that.
It's very similar to the zebrafish.
You mean you did it? You try some
There's-
pieces get down the no.
There's no data available. No.
Okay.
But I wouldn't expect a major difference in model performance.
Really different physiology and life cycle. Yeah. I will explain more about that. Yeah.
Okay.
Then when you it was a really good strategy to target a specific region of comparison between the different genome instead of going for the whole. And then why have you not looked at already between the fish genome which regions are highly conserved, and then do the study after? So here you look at in which region the methylation were stable. But instead of that, because methylation, this aging clock based on methylation is really species specific. So why you haven't looked at the conservation first, then after look at what there is in terms of methylation now?
This would be a good approach if you would have whole genome-wide data available.
Okay.
The problem here was it was 3 different settings. For zebrafish, we had a lot of data, but it was 3 different settings. So I know there are no exact same locations available. And that means I have to work in a manner where I want to conserve as much data as possible, with basically accepting the fact that I have to work with false positive errors. And this means that I start with trying to retain as much data as possible, and I think this approach, it has never been done before. This could be another approach. But this is, I think, an approach which worked in this case, and as I said, could be used in general epigenetic analyses where you want to compare not only data points among different species, but also within one species. Because you usually don't have the exact same data points, the methylation data points for all these specimens.
And in the fish, family, so is there, or did you highlight any specific region? I mean, when you sort.
I wanted to do a genetic analysis. I basically showed you 10% of what I did. I also had
Okay.
10 different models. I tried to simplify it as much as possible. But yes, I would love to look into the regions these methylation data points fall on. That's basically, I think, what you want to
Yeah, and
look at
in
the
the
function.
identity, what there is in terms of sequence.
Yeah,
DNA sequence.
exactly. What's interesting, which is something I am certain of, and I can show you, is that I talked about demethylation, hypermethylation, and hypermethylation. And what's interesting is that both models use the same data, but they don't give the same importance to each region. For example, in blue you have the chronological age model, which looks at the chronological age, and the relative age model. And these are the shared methylation regions the model used. And you can see that the regression, which is basically just the statistical model putting a weight on each of the locations and then sees, okay, this, for example, has a lot of weight, meaning that if you change the methylation value of this data point, the expected change in age is the greatest among these. And there are positively correlating ones, or positively regressed ones, and negatively regressed ones. And from that, I would look into what is SMR15, what does it fall on? I used human as the reference genome.
It will be my next question,
Yeah.
so because there is a lot of data in humans, so you have to
Yeah. Yeah, and I initially wanted to compare to the human clock or the mammalian, if there are conserved regions. I know there are some already, but I haven't checked the genetic importance or the function. That's something I would love to do, but I had to stop digging for results at some point because
You can go for
it was already, it was a lot of work.
Come back.
Alright, yeah.
No, it's okay. To you,
This
we
is
will come
really,
back
really
to it.
interesting for me as a marine ecologist, because we would like to age animals and even plants, but and this sounds like a really nice method. But when I looked at your correlations, I saw that the maximum age was like 12.5 years.
Yeah.
Yeah, and if you look at the data, it looks like the relationship is not linear.
Yeah.
So even if you put much older, do you think it will be linear, or maybe it's a curve?
My answer has to be a guess, I'd say, based on everything I read, based on every model I looked at, and I looked at a lot of models, I would expect this to be a linear relationship.
Okay.
But look at this species. When it's young, and I didn't do analysis on this because this would be on top. But look at this model, how it sees the snapper, which is older, but you can see in this area, it underestimates the age of snapper. In this area, it tends to overestimate. By just looking at it, visually, it tends to overestimate. But if you would basically make a line, I would expect a trend like this. I wouldn't expect a curve. Why? Because the relationship is linear. It's usually in terms of methylation change, you have a logarithmic relationship, meaning that in the developmental stage, the epigenetic modifications there's a lot of change happening, but then it tends to plateau and go to a linear relationship, sorry. And this is what I would expect here but that's a guess, of course. The only thing to find out is to...
In your introduction, you just show us some animals, groups of animals. Can this be done with plants as well?
Yes, there are epigenetic clocks for 2 tree species. I think. The problem is that I didn't want to include it in this case because epigenetic clock is a very broad term. In this case, we use methylation data for CPGs. In plants, it's usually CHH, the But also methylation data, but it works a little bit differently. How exactly, I'm not confident enough to explain, but yes, it exists.
Yeah.
And I'm confident enough. But
And when it comes to plants, what's the age there? Is it the age of the biomass, or it's the total age of the plant? Because you have biomass that is just 1 year old, and then you have biomass that's 10 years old.
Yeah.
That was a tricky one, maybe.
Is the issue with having hormones also?
Yeah. Maybe in some animals.
You should come back to animals.
It is an interesting question. The problem is it's not within my field of interest, because I'm interested in plants, but there's not much research been done on plants. In humans, for example, there's a lot of research been done. We know that different tissues and different cells have different ages. For example, nerve tissue ages slower, and also epigenetically, which is a hallmark of aging, the epigenetic drift, for example, ages slower than blood, for example, or skin, which ages way quicker. So I would assume, but please, I have no idea, that in plants it would be similar as of comparing humans to plants, which is not very
Really interesting. Thank you.
Coming back to you, Adina. I think other interesting questions.
Maybe curiosity question. So, do you know any animal that challenged this concept of methylation that do not have, for example, or start the life with full of methylation?
I would love to have your methylation data as soon as you have it for Nematostella. Please send it to me. I thought a lot about it. It's difficult because in fish, the aging pattern is different than in humans, for example. Fish are so diverse. There are the pattern itself looks differently already. So it's always a matter of what are you trying to compare with what. Of course, in my case, I wanted to look at conserved regions or conserved methylation sites. But it would be super interesting to know, for example, for Hydra or Nematostella, what it would look like, especially during the regeneration phase. What is interesting is that there's a lot of studies which also work on regeneration aging, and that in humans, for example, if there is a phase of regeneration initiated, there's a reset which is a pattern, an epigenetic pattern, which is similar to the embryonic state, meaning there's a reset basically when there's regeneration, which links, and I think that's beautiful, it links aging and regeneration capacities together again, yeah.
Thank you.
But yeah, give me the data, please.
Maybe I would like to conclude on something a bit more perspectives and large. So, maybe you have created a new way to measure age for fish. Not you and not the only one, but we are measuring age for fish with otholites for a really long time. Maybe we're wrong with that. What do you think will be the consequences of such discoveries? Not next year, but in 5 years, or what could be the consequence of that?
As of now, all the species where there is an epigenetic clock available, talking now, not in 5 years, you could use the tissue they used without killing the fish, so a non-lethal method, to accurately, extremely accurately and very precisely estimate the age. Way more accurate than using autologous data. I see this, if there's not another cheaper method upcoming, as the future of age estimation, not only in fish, but basically all animals we're interested in, and where we can link pattern with age data. In this case, with methylation data, but also there has been transcriptomic data, there has been even our gut microbiome was used to create statistical models to estimate our age very accurately. But epigenetic data has an accuracy and precision unseen of anything we used before. So I see in conservation, in ecology, in fishery science, I see this as the future, and it gets cheaper and cheaper and cheaper as we talk.
Thank you. Oh, we have a question.
Yeah, I have a question. First about methylation. I'm impressed and a bit jealous. But yeah, very impressive. Just following your question.
Gabriel, could you come closer to here, closer to camera?
I'm trying to do more or less the same thing, giving model, but with the lengths to guess the states of fishes. And you said that you don't have to kill fish to estimate the age. So that's wonderful for vulnerable species and yeah, in different cases. What do you think are the limits of this model?
In what sense? In what sense?
Because I guess maybe you are talking about linear, you don't know if it's perfectly linear. Thing like this, maybe mathematical issues, how to compare these values, because you have to predict this other value because you train the model, something like this. So have you seen some potential issues and limits with this method?
Okay, okay. Now I can give you a very general answer to this. Because basically you're training a statistical model, so the data you're working with has to be as accurate as possible. Meaning that if you use relative age, for example, that you're already, well, tend to make an error by just using the maximum lifespan we know of for a species, which is very likely wrong. So if the maximum lifespan data we have on the species is wrong, then your model will be wrong as well. So this is one thing. That the other thing is you need to know the age beforehand to train a model for a specific sample set. And this means that you have to have a sample set of a specific size. In this case, it was around 100 per species, which is a very good amount. This has been proved by a different study where they basically tried to simulate how many samples you would need to create a very good and accurate clock. But it also needs to be
Gabriel, 30 seconds.
It also needs to be well distributed, meaning that it should not be the case that you have one species where you have all your samples clustered on one age group, but it should be spread evenly along the age range, basically. And if you have these factors, and by the way, you can introduce length as an additional factor for the model like this, has been done already. Any factor you add, usually the better performing the model gets, because the more data available, the more the model has to work on. So
We have to conclude, and I'm sure we'll be happy to continue to exchange with a coffee outside.
Yes and no question.
No, we have to. Also by respect to everyone, so everyone has the same time for presenting and questions. Thanks a lot for the three of you. It was super exciting. Coming back at 4 in 50 minutes.
formed through the interaction of seawater, circularization, and magmatic heat source. In the Atlantic Ocean, we have the Mid-Atlantic Ridge. There is an abundance of volcanic activity and hydrothermal vent fields. In the deep sea, there are no sunlight for primary production, so the marine invertebrates that inhabits the vents rely on the symbiotic relationship between chemostatic bacteria, which the bacteria relies on the chemical energy from the vents and form as a primary producer. So the fauna at the vents are in species, and they are disciplinary species, and they are able to withstand the harsh living conditions. They are quite productive with low diversity and high abundance. Menesquen is located at the Mid-Atlantic Ridge, where the most dominant species is Azorius, and other endemic species like shrimp, limpets, and crabs. There was used to believe that the transition zone from hydrothermal vents and neighboring environments was quite sharp, but now we know that there's more transition zone within that. We have a lot of advanced technology now, so we can investigate and study the deep sea at a greater extent. But it also comes with interest in exploring the resources, especially deep sea mining, which is quite in interest at vents, with all the minerals at active and inactive vents. We also have a video of the advanced technology has led to more that we can study the area more with videos, especially, which is used a lot as it is a hard area to come to and study, and we can't use the normal methods. So the two main objectives are kind of two parts, which are spatial and the characterization of species distribution in transition zone from active to inactive hydrothermal vent field. And then a more temporal variation of the faunal assemblage of species Platypodilus, Azorius, shrimp, limpets, and microbial patches. So this is the study area. This is Menesquen, which is, I said, in the the Atlantic Ocean, and Menesquen is in the black square. The spatial variation study was done by two transects to see the distribution, the difference, along the transects in 2 different years, the yellow and the red one. The black dots indicates the active zones along the transects. And then for the temporal variation at fauna, at the vents, we looked at 2 different sites, cage site and white plain in Menesquen, and also here the black dots are the vents. So that are collections. For the first part, the spatial part, to look at the different species, I used the videos from 2012 and 2002. There we did video annotations and identify species at the lowest taxonomic level as possible, but it was hard a few times. That's bad lighting, resolution, and the videos were too far from the seafloor. And then did the principal content analysis. For the temporal study, I used video from 2011 and 17. I made a photomosaic image to get the whole site in one image, and then to find the with the reference size, we're able to find the relative abundance for each assemblage, which is here. I didn't affect any shrimp, so it's not included, but the different muscle assemblage are all muscles, but it's microbial patches covering muscles, only muscles, and then limpets covering all muscles. And then substratum, like anhydrite minerals, hydrogenous mineral sediment, and basalt. So this is the spatial variation from the 2 years. It shows that there was a group in both years that Bivalia, Teleosta, and Malacutrasa, and Teleosta was most associated with hydrothermal active vents, and basalt and hydrothermal active vents. With the distance of the closest distance, 0 to 100 meters. And then more sponges and corals were also to the furthest away with size 9 and tail 8 there, from 700 to 900 meters. This is the density map along the transects, which is in phylum and not class. Here we all can see that the mollusca was our highest density at the vents. Here. And also Cnidaria, Chordata, Arthropoda, and Esendermata was found along the whole transect near and also furthest point, with sponges the highest further away. Okay, so this is for the temple study on the fauna. Here is one of the white flame hydrothermal sites. The blue is the venting coming from chimney structures, and the light blue is just the diffusion zones straight from the seafloor. Here we can see that the changes, there was demolished in the biggest chimney structure from the 11th to 2017, and but also accumulation of new chimney structures here and more intense flows there. Also, the green is the microbial patches, which increased with also from 2011 to 17, and also the yellow is the limpets covering muscles. And also the basalt ended up covering was covered in sediment in 2017. Here you have the cage site, hydrogen site. The most active, the intense flow was in this area with, and also some diffusion points here and here at both sides. Most of it, the area is sediment, that's why this is so huge, there's no normal sediment. Also, there was no detection of limpets in 2011, which I could see in the video. And there was a more abundant, the assemblage was closer here compared to here with more space between them. Okay, so we know that the species Photinurgius azorius is endemic, and it was expected to see it, but other that was associated with vents was Malacustrasa, Polychata, and Teleoste, which was found in there. The Chachaeon ebontinectes is classified as Malacustrasa, was observed inside the vent, feeding and scavenging of the mussels and other vent species. It's an indication that it is less toxic vent at Manasquen, since they're able to actually move in and out from it, and they're not endemic species. The environment close to the neighboring the hydrothermal vents are coral gardens and sponges along the whole transect. Hexagralia was found along the whole nearest vents. Has previously been studied that in the food intake of the area from 20 to 500 meters a distance, and they found vent associated species. So we can indicate that they are in the transition zone, benefiting from the hard substrate as well as the vent, as they are sessile feeders and they settle in one place where they rely on the current. The vent fluid and temperature is not fixed at the hydrothermal vents, and they will vary quite at a small temporal and spatial scale. So there's a lot of fluctuation in temperature and the minerals there. You can see that also in the chimney structures that got demolished and changed the direction of the fluid. There are also accumulation of new ones, making it able to making a new era able to live for the mussels. There were also found a gradient of limpets and microbial patches, which means that the closest to the shimmering, you can find high abundance of limpets and microbial patches, but they will gradually decrease further away from the vents compared to the mussels. So we know that there's a sphere of influence where we can find species, non-endemic species, benefiting from the vents. Here we found Coralia. Also the assemblage follow the fluid flow and will settle where it's most suitable as they can move, making it an advantage, the same as microbial patches. Further studies would be advised at more species-specific traits to actually see what kind of traits make it possible for some species to live closer and actually take advantage of it. And also look at the temperature and more chemistry, not only in the vents, but further expanding the area. But it's not possible on a video. So, and also deep sea mining, which we see now, how much not only if we infect and impact hydrothermal vents, but it will also impact the area around. Yeah, and reference. I think.
You used the 13 of your 15 minutes, so there was some time left if you had anything else you wanted to add to it, but yeah. Could I ask the members of the jury to start the discussion? So Fabio, Lars, whoever wants to go first.
Look at me go first, Lars, because here we are really in a field which is a bit out of my expertise. So.
Thank you for this presentation. I have a question about the method and also results, new interpretation, also about the introduction. What is the context that triggered the question that you wanted to solve? What was the unknown that needed to be investigated?
Well, now there's not that much news, but it was more like what species that were in the transition zone.
Because I guess you have very nice results. In 2 years, if I understood well, of the same location, the same reef or the same event, and, yeah. And you are trying to find some similitude or
for the second part, the same information look at how they changed over year. Yeah.
So what was the question? What is lacking in our knowledge? Because it's ecosystems which are very deep, very hard to investigate. So to a sort of money to have a result there, so you need to have a question that you really want to solve.
Yeah, what species are closer and more can withstand like the actually benefit from the hydrothermal vents, I would say. And if
But
there's
could you
like other
answer Could you answer this question?
I would say no, because I only use video. But I would say the observed one is the hexacoralia.
Yeah.
And the sponges.
I was wondering, the ROV is not equipped with temperature sensors?
Yeah, but I only have videos.
Okay.
Yeah.
The data was not available.
No, yeah. But that would be.
Okay. Can you go to this, to your PCR results?
Yeah.
So you call that spatial variation, but when I look at the two PCA, I don't understand why you call it spatial variation.
It's more because I try to divide the twos in like spatial and temporal parts. So it's more like, yeah.
What does it show? Because I don't, from the place I am, I can't read the arrows. I can't.
Okay, so here is hydrothermal, which is like active area, and here is basalt, just like the substrate type where they were identified. And the site here are the distance, so it was divided in intervals, 0 to 100, 100 to 200 to 900, to get where they were observed, like from the distance from the active area.
Okay.
Yeah. And these are the different classes. And here you have basalt hydrothermal and basalt hydrothermal, which is gradient from active hydrothermal is the active area with basalt.
Okay.
The transition is, yeah.
That's from the same location?
No. So there's a few different translates, which is like here. One goes like that, and other goes like that. So it's not the same.
Ah, it's a data along a transect.
Yeah.
Okay. And so if you go back to the PCA, can you compare the two? Would you say that the organization in 2012 shows different things from the organization of 2022?
Well,
would the species or the groups which are typical of active sites be in relation to the descriptor, be in the same location on this plot, or?
Well, I can't really compare since they're like not in the same place, but you can see kind of like the trend of which ones are at hydrothermal and which one are further away from the events.
Okay.
More trend, yeah.
And then maybe I have another question is about the density maps that you are showing maybe just after. So how deep are we here?
Around 800.
800 meters.
Yeah.
How can you get the GPS position there?
The dive track.
The
dive track.
What's
So
the
they included in the from the cruise.
Yeah, but how do you know?
Well, it's on the camera. Yeah. Going up.
There is a GPS
Yeah.
on the ROV.
ROV, yeah.
Yeah, but the GPS is not functioning at 800 meter depth. I don't think so. The signal.
But you have positioning on the ROV.
Yeah,
but
so
it's not GPS. Yeah.
So I think it's usually how they do it. They put balise, buoys, I mean not buoys, but things anchors on the floor, and then they know the exact position, and then they can measure the track of the ROV between those four balises. So I don't think it's a GPS, but I'm really impressed by the resolution that is providing here. Yeah, maybe because it's concept, a better way of presenting this data set would have been along XY plot. There we would see up and downs. Here with the colors, it's more difficult to see. Okay, thank you very much.
Well, my question is, once again, more or less nothing of this environment. So, a general level, okay, based on the literature, how much do we know about the spatial and temporal variation in this type of ecosystems, especially this temporal variation? Something that would be interesting to me to understand. Do we know if this ecosystem change in time or not?
Well, yeah, it's a lot of change since it's not fixed, so it changes a lot over time. And there's been studies done like how you're talking about just events. There's like how far they are usually compared to the temperature and the chemistry. But I would say from going from vents to other environments, there's not necessary here. And
Oui, moi aussi j'en ai. Fabrice,
No, okay, I was wondering if there are other studies with which for other hydrothermal vents in other parts of the world, which you can compare your results.
There's some, well, this is quite a shallow one. So it's usually there are like 350 to 400 degrees. This one is 300. And yeah, less toxic. So, but we have this Pacific. There's a lot of vents, but there's tube worms, most of them. They're also around here, like further down here, the lucky strike, which also been a lot compared to Venezuela. It's a little bit deeper, but yeah.
I wonder, are there any general patterns that are known?
Well,
some kind of animal groups that tend to be dominant in general in these environments.
Yeah, well, the mussel is dominant, especially for the lucky strike. But also when it comes to the other like shrimp, you can find another shrimp dominant there. This one is more dominant with the yeah, one of the Marpidurus, I think, yeah. So.
Thank you. I have a question about, can you go back to the PCA? Because I'm still trying to understand it, actually. Even after Lars discussed it. Can you explain to me just clearly in any one of them, what are the sites, what are the rows of the input table, and what are the columns, what are the variables? Because I'm seeing points for sites, I'm seeing points for taxa, I'm seeing arrows for basal, so I'm not understanding what's going on here, actually.
We have the columns, is the class.
The columns are the class?
With the abundance.
Okay.
Counted. And then I calculated the distance with in intervals. So first the distance, and then we put in intervals, 0 to 100 meters from the nearest active end. So that's the site. So 1 is the closest one, and then to 9 is the first.
Okay, but in the input table you use for that analysis, it's one table, the rows are what?
Rows are the intervals with the intervals, class, and then abundance.
I'm not understanding. Because normally you would have the sites as the rows and then the taxa.
Yeah, it's like that. Oh, sorry.
But then why are the taxa not the arrows in the graph?
Because then I put on the environmental variables.
So did you mix the environmental variables?
Just put on after.
After, okay. Now you need to tell us that, because when if I see like, oh, I think she used both in the same analysis.
No.
That doesn't make sense. Okay. And did you use any kind of transformation on your taxonomic data?
Hellinger.
Okay, so yeah, you need to tell us these things, because otherwise, again, we don't understand the graph. And we're like, maybe there's something wrong behind it. So yeah. I know, Lars, I'm sure you have more ecological questions than the technical questions.
No, it's a technical question.
Okay, also.
About the community, the different types of community that you are defining. I was wondering if you were participating to this work, or if it was given to you. So did you have to video screen out of videos or?
Did you do the annotation yourself?
Ah,
The
yeah.
video? Yeah.
Yeah. So I looked, had got all the video, looked at it myself, and then when I identified, I did, and then heard with the professionals if it was right. Yeah.
Okay, because this 3D reconstruction that you're doing, it must have been quite a lot of work.
Oh, yeah.
And there is no specific software to help the decision on the classification.
No, I use Beagle software. Yeah. Or no, that's it. And for identification.
Okay.
And for the
all the annotation is human-made.
yeah.
There is no machine proposition to the annotation. Okay.
And
But people shift it.
Yeah.
So, yeah.
and how do you define then the habitats? Is it you again, or is it an algorithm that takes the annotation to decide this is such an habitat and this is another one?
Previous studies from that area, and then yeah.
But then it's a decision of the human in front of the.
Yeah.
Okay. All right. Any? Go ahead, please.
Maybe
No, we have time. And you can, if you have questions, please raise your hands. But you have a few minutes left. Lars.
Okay, so you are mentioning in the title, in your introduction, and in the conclusion, deep sea mining.
Yeah.
How do you relate your work with deep sea mining?
It started out to be supposed to be a much bigger part of this, but it really didn't. Well, I wanted to see how first, because there's a lot of interest in the active areas, so I want to see if we infect, no, I'm sorry, the hydrothermal vents, and if we infect that, how will it affect kind of the surrounding, and how much does they benefit from it? So I want that, yeah, I include that in there.
So, but what kind of deep sea mining is expected there? Will it be beneficial? Will it be... Can
No,
you elaborate on it?
I'm sorry.
Can you elaborate what was the initial goal of the study? I did not get it.
To look at the spatial variation and
Characterize a spatial variation. Maybe.
Yeah, but where's deep sea mining?
Why? Because
No, but is deep sea mining expected for this zone or for another zone? Or?
not in this area, no.
Okay.
Because this is kind of in an MPA, but it's expected to be further south. Yeah.
All right.
Thank you.
All right. Thank you very much. Now call Sarah to stand. Yeah, you are online.
So be careful.
Yeah, I think.
Je peux le poser le.
Oui. Yeah, the escape.
Escape.
Yes.
Don't escape. Try not to.
No,
Sarah, whenever you are ready, you have 15 minutes.
Thank you very much. Good afternoon, everyone. And good afternoon to also the people that are on the live stream. I will present you today my thesis defense that I conducted in southern part of Chile about the sexual life of dolphins. As you all may have heard, dolphins have a quite active sexual life, but in reality, it is rather complicated to study because mating behavior in free-swimming cetaceans happens, of course, underwater.
It is opportunistic and can be very brief. It can be only a few seconds or a few minutes. And apart from all of that, most species of cetaceans avoid boats, making it really hard to spot. And if they don't avoid boats, they're most likely gonna modify their behavior, making it even harder to study mating behavior. But a new tool is developing and it's increasing in the use of cetaceans research, which are drones, also known as UAVs, unmanned aerial vehicles. Thanks to them, we get a whole new perspective over free swimming cetaceans, and we can see underwater behaviors that we were not able to see before from land or from a boat. Nevertheless, studies with drone-based studies based on sexual behavior and mating behavior of swimming cetaceans is rather limited. So this is what we did. We used drones to characterize the mating behavior of an understudied species in southern Chile, which are the Peel's dolphins, Sagmates australis. They're one of the most frequently observed species of all cetaceans of southernmost South America, but they are among the least known species in the dolphin family. In fact, the age at sexual maturity was and is still undetermined because of the low numbers of specimens that could be analyzed. The sexual anatomy has never been studied. The reproductive season is believed to be between February and March, but was only mentioned in a paper done in 1997. And for reproductive behavior and mating strategies, they are also completely unknown. So the aims of this study was to determine the group size and composition of mating groups, to describe and determine the frequency of this behavior and the sex-specific sexual behavior for females and males, and finally to determine the mating tactics of females and males in Peel's dolphins. So we conducted our study in the southern part of Chile, in Punta Arenas, on the western coast of the eastern arm of the Strait of Magellan, along a 50-kilometer coastal road that connects the city of Punta Arenas to Fuerte Bulnes in the south. We conducted land-based studies with at least 2 observers that scanned the sea at all time with binoculars, and as soon as one pod of dolphin was sighted, the drone was launched and we conducted continuous focal follows over the mating group. This was possible because the species is very coastal, as you can see in this picture, with a pod of 11 dolphins. Apart from the fact that the study is a land-based study, so that it is non-biased, we always made sure to fly the drone above the pod of dolphins, always at an altitude above 30 meters, above or equal to 30 meters high. And studies on drones are often limited by battery life. It is why we use 2 drones. 250 pods of dolphins, and we managed to follow 171 with the drones. This encompassed 40 hours of drone recording on dolphin pods. And of those 40 hours, 13.61% were of only mating behavior. So it totaled to 5 hours 29. And that is what we will focus on. So I put a reminder of the aim we are trying to reply to during the results. So we focused on these 28 mating groups and it's 5 hours 29 of recording. We overall, throughout the whole study, the mean group size of the pods of dolphins was 4.89. But we can see, well, that the mating group size is actually just slightly over that number with 5.21 dolphins. But there's a difference with the mating group size and the mating individuals, which are the dolphins that actually participated in the mating group. Because in mating pods, not all the dolphins participated in the sexual behavior. It's something that we can clearly see here, whereas the mating group sizes range from 3 to 12 individuals. There was never more than 6 dolphins participating in the sexual behavior. As for those compositions of those mating groups, we can see that there were up to only 2 females participating in the mating behavior, but that most of the time, 73% almost, was formed of only 1 female. For the males, it was a bit more different. We can see that they were mostly 2, but could also be 4 and went up to 5 males in a mating group. For the second aim, which was to describe and determine the frequency of sexual specific behaviors, we created an ethogram with 31 sexual behavioral events that we categorized into females, males, and behaviors that could be showcased by both sexes. And females, we further categorized them between evasive behaviors, receptive, and ambiguous. Here are some examples. A really clear one for males is the averted penis, penis out. And for females, it could be evasive behaviors such as the body roll, when the female tries to escape males that want to go into the copulatory position, or females that swim fast. Here we can still see the momentum on the picture of a male chasing a female. So during those 5 hours, 2,844 sexual events were recorded, of which 21% were showcased by both sexes, 46% specific to only males, and 32% were specific to females, with a huge majority of evasive behaviors. Of those behavioral events, a lot of them were copulatory position, but only half were considered successful, where there was an intermission of more than 3 seconds. Each successful copulation had a mean intermission duration of 4.38 seconds. And for the third aim, to determine the mating tactics of females and males, we based ourselves on previous studies on other species. So where they observed that analyzing the copulation rate per minute per male in groups with increasing number of males, the copulation rate per minute per male could either decrease, which would mean that there's a competition between the males, or the copulation per minute could increase, which would mean that the males cooperate between each other, as was seen, for example, for bottlenose dolphins in one population. So we did that in this study using generalized additive models. We analyzed the copulation per minute per male in relation with the number of males, but also with the sex ratio, group size, and mating group size, expecting, as I said, that there would be a decrease or an increase in the copulation rate. Nevertheless, it did not do that. It did not decrease and it did not increase, which meant that even if there was 1 male or 5 males in a mating group, they always managed to have the same copulation rate per minute per male. So we calculated the copulation rate overall, and this time not per male, and unsurprisingly, it skyrocketed whenever the number of males in the mating group increased. And that, coupled with the fact that we know that the mating groups have very few females, mean that as the number of males increase in the mating groups, there's an increasing pressure on the only 1 or 2 females that are present. which led us to calculate the evasive rate of females and coupled it with the sex ratio. And as we had expected this time, as the number of males per female in the group increased, we see a significant increase in the evasive rate of females. This, coupled with all our observation, led us to conclude that males use scramble competition as their main mating tactic. But we propose a revised option since males in this population do not seem to compete. So we propose a non-competitive scramble competition where males will try to copulate with as many females as possible without actually interfering with each other. For females, it's a bit more complex, but they seem to be using signal discrimination, evasive behaviors, and multiple matings, during which they evade males, and by doing so, put them to the test, put their agility to make sure that the males that actually get to them have actually good agility, speed, and that those are traits that could be inherited by their offspring. And at the same time, they evade the males, which means that the weaker ones will not be able to keep up. So I put again this slide that I put at the beginning. And we are now very proud to say that we can confirm that the reproductive season is between February and March, although we hypothesize it is much longer, but it is during the austral summer. And the reproductive behavior and mating strategies of this species are now known. So during these 5 months, we were able to characterize the mating behaviors of these species. We were able to say that the groom numbers and compositions are, they're most commonly formed of 3 adults, 2 males and 1 female, that there's very rarely juveniles among the mating pods, and that the mean size of the mating groups is 5 dolphins. We were also able to establish a frequency and know that they spend 13.61% of their time reproducing, which is quite significant, and built the first ethogram on sexual specific behaviors. And finally, we were able to give specific mating strategies to males and females, where males use non-competitive scramble competition and females use signal discrimination, evasive behaviors, and multiple mating. These 5 months of data collection were very intense and allowed us to collect a lot of data, which is encouraging for the future for long-term study, and hopefully be able to include seasonality within this study. Thank you all for your attention.
Thank you, Sarah. You had about just a little bit over a minute left of your time, but that's pretty good. So again, I turn to the jury members to start the discussion. Fabio or Lars, whoever wants to go first.
Okay, they're very interesting. Obviously, dolphins they know how to enjoy their lives.
That's really
I was wondering what is the height of the drones that fly above the. Okay, well. I understand. I was at the other presentation, so I understood about 30 meters. That's right.
30 meters, and we could go up to 130 meters according to the local regulations. So it was, and the drone had a.
You really can recognize the. Way to recognize the sexual behavior.
Of course, the video I put was one of the best visibilities we've ever had. I would have hoped that the entire 5 hours are with that visibility, but sometimes we had worse visibility due to waves or the sun or just the climate. So we categorized them and actually removed some of the footage because of too low visibility, which could not allow us to, because we're really analyzing each behavioral event. So we removed and only kept the best visibilities.
Okay. Look, from this point of view, is there any pattern in relation to the season or different times of the year.
So it's the first study on sexual behavior. The previous study that mentioned it only mentioned that during February and March they saw an increase in the citing of this behavior, but it has never been quantified. We believe it's really during this, the austral summer, because we saw neonates, and we believe the gestation is of a year. But of course, I would be really curious to go back and actually keep on looking at the sexual behavior, because they have also non-reproducing sexual behavior, which is just for fun. So to quantify it and see the differences in what we can see. That's why the lithogram is a good baseline, because we will be able to compare maybe the frequency of some behaviors compared to others.
Just one last thing. Okay, one of the first slides, okay, you mentioned that you defined the sexual behavior opportunistic.
Yeah.
So opportunistic, what does that mean exactly? They just jump on each other?
Well, it's that you can't predict it. You don't know whenever a group is going to start having sexual relationship.
Yes.
So it's also something that we would like to maybe study. It's the trigger. Doing for Markov chains, for example, to see what kind of behaviors, for example, maybe it's what we saw, but it's not at all confirmed. It would be a hypothesis. They join in larger groups to forage and feed, and then scatter into smaller groups and start having sexual behaviors. It's things that have been observed in other species. They said that after a successful hunt, there's a lot of chance that there's going to be reproductive behavior.
Okay, thank you.
Thank you, Fabio. Lars?
Yes, thank you for this very original work, both in terms of what you have been studying and also the place is very far away from here. It's like another planet.
It is.
My question is, why is it important to assess the behavior, sexual behavior of this dolphin?
Yeah, it's a very interesting question. So it's a very coastal dolphin. The entire study period was really just land-based, and we saw an incredible amount of groups. So around Punta Arenas, around the study area, there's a really big city, which is Punta Arenas, and there's a huge anthropogenic impacts that are linked to that. There's urbanization since the study that I mentioned in 1997 that started taking interest in dolphins was also around here. And what we can see is that now there's so many more houses built all over the 40-kilometer road, which before had no houses. Now there's so many. So it comes with pollution, urbanization, anthropogenic impacts, and an increasing whale watching tourism, which comes close to the dolphins. There's no regulation, and the dolphin has a super approaching behavior, really strong approaching behavior, starts sleeping. And so if it is really important to target when it happens, so that we can maybe advise maritime traffic to not go around, close to the coast or close to reproductive areas, because, if they stop reproducing, then the population might decline and everything. So it is really important to study them.
And I was wondering if the goal is to manage this, the population. What data would you need for these dolphins? What would be useful? If the objective is to clearly manage the population and try to even predict change in population due to specific pressures, what other data would be useful?
So there's seasonal data, of course, but also because we know that in the winter season, there's less dolphins presence, and we do not know where they go. We do not know if they migrate. We do not know how many they are, if the 250 pods we saw were resightings or not. So we also started a catalog, photo ID catalog, because they don't know if it's a resident population or not. So yeah, in the best case, I think we would love to be able to put tags on the dolphins to see where they go during the winter season, where the moms go to have their neonates. Would be very interesting.
I was wondering, so it's a group, and you just said that sometimes they gather, sometimes they are dispersed. How well do they know each other under the water? And do they communicate also using, acoustic?
So there's a very high rate of fission fusion. Well, fission and fusion. So a pod would stay of 8 individuals, and then 4 would leave, and 2 of another pod would join. So it was a very dynamic and fluid society. So how well they know each other, I am afraid I don't know. But it was interesting to see, for example, we had a dolphin that had a very distinctive mark because he had a plastic around his neck. And it was very sad, but it enabled us to identify him really quickly. And we saw him more than 6 times, I think, throughout the span of a month, around the same kilometers, which are like to the kilometer 36 to the 48. So it kind of shows that it's really important to keep on doing the photo ID catalog that we started, that a student is reviewing now, the pictures that we took on the field, and to see if actually we see a lot of resightings or not, and have more information on that.
And do they use acoustic?
Yeah, sorry, I forgot to answer that. They do use acoustics. There's the studies that mention field dolphins are very like a long time ago. But recently, actually last year, there was one published about their acoustics. So they're starting to see what they use. So actually, acoustics would be also interesting to put some hydrophones and see how they communicate and link it to some of their behaviors to see.
Okay, thank you.
Thank you.
Yes.
Great presentation, like such a great scope and scale of work you've done. I have a very specific question related to the evasive behaviors of the females. So I think at some point you said that there was about 95% of the behaviors of the females were evasive. And you also mentioned that there was an increase in the evasive behaviors of the females when there was a great number of males, like sex ratio. How is it that you were able to quantify and sort of navigate this difference when you already had a 95% chance of evasive behavior?
So because in the evasive rate of females was most high when there were more males, but so because we had 2,000 behavioral events, and of those 2,000 we categorized between sexes, females and males, and each of the females' behaviors were categorized into evasive or receptive. So for example, a body roll would clearly be evasive if a male was trying to copulate, and females showed very rarely, but they did, receptive behaviors, such as presenting their ventral area towards males and actually actively refusing some others. So it shows that they have a preference in the groups, but of course there's also a compound that is that they're also passive. If they're not doing anything, it's neither receptive or evasive. So it's difficult to quantify this like receptive behavior, and we quantify more the evasive one because it's more clear to us. And most of their behaviors, of the behaviors we described in the ethogram, are evasive because it's what they do. So in groups with lesser males, females will do less evasive behaviors per minute, whereas in bigger groups, they did a lot more in the span of a minute.
So per minute?
Yeah, exactly.
Yep.
You talk about all the diagrams. There is a behavior that you attribute to both.
Yeah.
Do you have an example of it?
Of the histogram? Yes. It's at the entire end. Here is an example of the histogram with the count of each behavior. So in the order of the most common one to the least common one. So for example, for the both sexes, the most common one was the successful copulation. For males, it was the inverted swim, and for females, it was the body roll.
Just a quick question for the photo ID catalog. Are you doing it by hand or are you using specific software?
So we wanted to use a software because we had some experience in Montenegro, but with bottlenose dolphins. When we used a software that is called Discovery. So we wanted to try to use the same one, but because there's no database on Peel's dolphins, you need to teach the software, and the best way for now is just do it manually. Yeah, and we contacted Argentina because so the dolphin can be found on the coast of Chile and Argentina. And there's research being done in Argentina and trying to bring all these countries together so that we share the knowledge on, well, the little knowledge that we have on Peel's dolphins. And they were all doing it manually since 40 years, and they have a population of 40 individuals, and the woman is super passionate about it and actually draws each of the fins. So it's passionate, really, well, incredible to talk to them, but for now, no, it's everything. But we might, once we have at least like 30 individuals recognized, put it in a software so that it can go a bit quicker.
Okay, well, we're going to talk about it later, but okay, I'm interested in this.
Vincent, please.
Yes, thank you for the presentation. It was cool. I had more technical question. I feel like drones are a very good technological tool for research, but I guess it's very dependent on weather conditions. So yeah, when it's wind or rain, could it be a problem to make like time series observation and database?
It's definitely a problem. But I'm gonna say if we manage to get so much in Punta Arenas, which is one of the windiest places I've ever been to, it was quite incredible. We were really lucky with the weather. The drones cannot go over if there's more than 40 km/h wind. And it's unadvised with rain. But of course, because of our location, we really push the drones to their limits. But yeah, it's something that needs to clearly be taken into account. So I think every morning we were looking at the weather forecast exactly, and seeing if we could go or not. But it worked out. So yeah.
And thank you for the presentation. Just two very quick questions before we let you go. The way you're talking about it, it seems like you're continuing to work on this project afterwards?
It's something that we are thinking about still, but we would clearly like it, yes.
Okay, excellent. And can you go back to aim 3? You had a graph of, I think it was evasive behavior of females against number of males.
It's not against the number of males, it's against the sex ratio.
Yeah.
Which is...
Yeah, okay. Still, I'm not convinced by your interpretation. And I'm wondering, this is a linear model, so you have the variance is very uneven all along the response. So that's a problem for such a model. And when I look at that, I see two kinds of behaviors. Maybe you don't have enough data at this point, but maybe there are two types of behavior in the females, and maybe is it the same individuals always up there and others always down there? That could be looked into, because this split is very strange to me.
It is something that we further want to dive into. Actually, being able to track specific individuals, because I did it manually. So for males, it's a generalization. So we did females and males because it was too time consuming to do dolphin 1, dolphin 2, dolphin 3. So it's a generalization. So of course, there could be a lot of differences between females.
You're right. You don't have the kind of data. Look at
But
that.
really interesting. And,
Yeah, that might be something. Anyway, thank you very much. Super. And to close this day, we welcome Chloé Rose. You need to ask that with our gentle and nice room operator. I don't know.
See it. Oh, it's so small. It is. It's very small. You can see it on the wall over there and on my hand, but not really on the LED screen. Oh well, it's okay. I have arms. Where's the best place to stand so people online can see? Oh, thank you.
Yeah, in front of the other screen.
Thank you, Rich.
I think if you're in front of the screen.
Center screen.
It's okay there? Okay. There's a lag between reality and the stream, so I cannot see you now. No, it's because there's a lag. You have not come into that time frame yet. Chloé, you have 15 minutes anytime you're ready. You start.
Thank you all so much for being here today. My name is Chloé Rose Combero, and I did my thesis just down the road at the LOB in Villefranche-sur-Mer with Maria-Louisa Pedrotti and Annalisa Lacerda on harmful microalgae living on marine plastic. So getting right into it, in a somewhat intuitive but disappointing relationship, the more plastic that is produced each year, the more of it ends up mismanaged as waste in the environment. And when we say, oh, we're estimating 1.7 to 4.6% of that plastic ends up in the ocean, that doesn't sound like a lot until you consider the fact that we're talking hundreds of millions of metric tons of plastic being produced each year. So as that amount increases, so does the pollution in the environment. And by fraction, if not by mass, the greatest amount of that plastic pollution is microplastic, which are smaller than 5 millimeters in size, half a centimeter. They're nearly ubiquitous in the marine environment. And they pose a sort of unique threat of ingestion and bioaccumulation because the smallest organisms in the food chain can consume them and indeed have, like copepod, zooplankton, they're eating microplastics. And they even will preferentially eat a microplastic that has a biofilm on it, which is known as the plastosphere because it smells like food. So what is the plastosphere? It is this incredible microcosm of microbial diversity that lives on the surface of even the tiniest plastics in the environment. In this picture in particular, no, we can't see. So we see these big kind of boxy guys crossing the screen. Those are macrophytes. We've got some pennate and centric diatoms. We have a little coccolithophore in here and loads of bacteria. There's a lot going on in this SEM image, which is 1000x magnified. It's a biopolymer that was incubated for 30 days in the season of spring in Villefranche during the season 2021. And of the diversity that lives on microplastics, it's known that since around 2000, we've been finding potentially harmful microalgae living as part of this community. So a potentially harmful microalgae is a taxa that contributes to the formation of harmful algal blooms. And these events take 2 primary forms, though of course it is a spectrum and there can be combinations of these 2 things, which would be terrible. But so the high density bloom, that is what sort of gives the HAB its nickname, the famous nickname of red tide. They discolor the water, they create mucilage that can fog watercraft, fishing gear, fish gills. They create an anoxia after their mass mortality that can create huge die-offs in fish farms and aquaculture, but also just in the habitat at large. And then the toxic side of things, it's a bit harder to see visually that this type of bloom is happening because very low cell counts are required to reach a harmful effect. And different strains, so diversity within the species level, different strains of a confirmed toxic species might not be toxic or will produce different compounds. So that's kind of where this potentially caveat comes in. We can't say this is toxic, even though we've seen them creating toxic blooms before, there's a lot of diversity within a species. Sometimes we only know a toxic bloom has happened after people start eating and getting really sick from contaminated shellfish and fish, and this can sometimes, unfortunately, be fatal. So it's a quite serious problem. It is a global issue. Just in the way that plastic pollution is happening everywhere, so too are HABs. They're occurring more with time, and people aren't sure if that's a reporting bias or if it's due to climate change. There's a bit of an open debate on that. But the forces that are moving small particles in the ocean, they're acting on biological particles as well as man-made ones. And so when HAB species are aggregating in a low renewable environment due to tidal action, wind, or waves and currents, plastics are ending up there too. If the plastics act as a reservoir for these harmful species, they could be potentially a vector for moving them around. This could extend their range. Or they could just be holding on to them, and that could extend the duration or the strength of HAB events that are already ongoing. I should have brought water up here. We wanted to know, will this community inside the plastosphere, will it follow the general trends of the broader microbial community that we've seen? So we know they're there, and generally many studies on the plastosphere have confirmed that geography is the biggest contributor to variance within these communities' compositions. There are conflicting results about polymer influence, whether or not influences richness, community composition, either, neither. The jury is still in debate. And so our questions are, will our experimental categories influence PHM? And that includes site. So we want to know if there are regional differences in diversity. Will we even have these taxa in our samples? If so, where will they be? We hypothesize that we will find potentially harmful microalgae in all of our sites because they have been reported in our sites previously in the literature. So we're expecting, okay, we're gonna find some, fingers crossed, although it is of course negative. We think that the greatest contributor to their variance and presence will be geography, and then within geography, it will be seasons per site. And we think that polymer type will influence the richness, but not community composition, because we have a very special type of polymer on board that has done this in the past in the literature. So because it's a global issue, we wanted to take a global perspective. The data that my project is using comes from the JPI Oceans Microplastics project, that logo in the corner there, which has dozens of partners throughout Europe and all around the world. My data comes from 5 sites. There are 3 in the Mediterranean, 2 in France, 1 in Italy, 1 up here in Ireland in the North Atlantic, and in the South Atlantic in Brazil. These are the sites that I have sequencing data for, but there are, of course, more partners than just these 5. This map is a chlorophyll concentration map for autumn in the year 2021 when the sampling was done from the Copernicus database. So as I mentioned, this data comes from the JPI Oceans Microplastic Project. It was collected from 2021 to 2022. So even though this part happened before I came on board, I want to run you through it a little bit, the experimental design and the data generation, so that you can sort of understand the type of data I was working with. So it is an incubation experiment. It took a whole year to complete in all 4 seasons. Plastics, which are the samples themselves, 1 centimeter squared, they were put into this sort of perforated cage incubation system, placed in the water at the start of a season, and removed at 7, 30, and 90 days without replacement. They weren't put back in. And then at 90 days, the new set was put in for the following season. Then on-site DNA extraction following a standardized procedure was undertaken by each of the partners, and they sent away those samples for PCR amplification of the V4 region of the 18S RNA gene, and for next generation sequencing at a company in Canada, which is here in the corner. The amplicon sequence variants, which are the result of that sequencing effort, they're called ASVs. They were classified taxonomically using PR2. And so this is where I come in. My work started with a massive literature review where I wanted to know what harmful microalgae were out there and which ones had already been reported in the plastosphere, because it's important to know what's already been found. For the purposes of this study, the potentially harmful microalgae are being considered at the genus level because we have amplicon data, and genus level identification is much more certain than species level. And as I mentioned before, there is even variation within a species. So even if we had confident species level ID, we wouldn't know for certain if it was a toxic strain. So we're considering if it is a genus that has a species in it, which has confirmed a report of a harmful algae bloom, it's a potentially harmful microalgae genus, and that's the scale of the data we're working with. After creating the data filters, filtering that down, reconfirming the ID with NCBI blasts, and cleaning the data, looking for erroneous reads and samples with too few reads, the data was made binary. So we're only working with presence-absence data because the sample type of sequencing and the target taxa have a really broad range of gene copy number for the 18S gene. So dinoflagellates in particular, they can have many orders of magnitude different in the number of these genes, so it would be really hard to have very certain abundance data. So we're using presence data just to keep that in mind. All the figures we'll see are presence. My analysis, I wanted to know some things about the proportions of the samples, what was colonized by PHM, what was not, and a lot of my study focused on the diversity that was present. So for our first result, we have on the side, the blue side, I don't know left and right now, we have the proportion of samples of all successfully incubated samples and sequence samples that did have harmful algae on them. In the Mediterranean block on the top, we see that there was a much higher proportion that had PHM present, and in Galway, we have a really, really small percentage, even though it has the second highest number of samples overall, which is interesting. That site in particular is hard to see in the map, but it's at the mouth of a river emptying into a harbor, so it had a strong freshwater influence, which could be part of the reason why there weren't many marine harmful algae species there. Rio Grande is somewhere in the middle. Over here on the pink side, we have the amount of all the microalgal ASVs inside the samples, which were positive for at least one PHM sequence. And we say, okay, of these sequences that we collect from each of these sites, how many of them were harmful? And with Naples and Galway as our extremes, both of these sites had pairwise significance with all the other sites and with each other. And again, Galway, very interesting, didn't have very many harmful sequences at all. When we're thinking more about the samples, we were looking at the polymer type, and we have, I should have better explained what the polymer types were on the method slide, I'm sorry. So just to talk you through them really quickly, LDPE is just a traditional fossil fuel polymer. PLA is a biopolymer made in part with plant starch. PPPC is a recycled fossil fuel polymer. So we have slightly different polymers for each of the three. And when we considered overall them as a group for all time periods, PLA far and away had the highest richness, much higher than the two fossil fuel-based ones. I wanted to break this down a little bit and look at it further, so I broke it into the timestamps of 7, 30, and 90 days. And in this situation, 7 days and 90 days for PLA still had higher richness, and it had the second highest median richness in 30 days. There were some pairwise similarities as well, or dissimilarities as well. And it's very interesting that PLA had the greatest ASV richness because in a previous study, this was confirmed for both prokaryotic and eukaryotic communities. When we want to move forward to thinking about the beta diversity, about the community composition, with a principal coordinate analysis based on a square root transformed binary Jacquard dissimilarity matrix, we only had one season in common between all the sites. So if we want to compare the sites, we need to kind of control the seasonal variation. So this is all sites in autumn, and we've got an obvious significant beta dispersion test. We can see that Naples has a really big spread compared to all of these sites clustered in here, but we can also see very clearly that the centroids of Toulon and Naples are very different from the other sites. So this was quite interesting that Toulon and Naples were significantly different from all the other sites. When considering community composition in the seasons, we have 5 sites. 2 of them, Toulon and Galway, did not have sufficient data for testing. Villefranche was not significant, so I'm sharing the interesting significant ones with you guys. We have Naples, where autumn was significant against all seasons, and Rio Grande, where there was a pairwise significance between the 2 available. Autumn, I wanted to point out over here, it has sort of a gradient from the shortest incubation time, middle incubation time, and the longest incubation time, which is clustered here. It's quite interesting. And of course, it would be awesome to be able to study the other sites further. This one is a lot to look at, but just to help orient you in the figure, it's still ASV richness we're looking at. It's just aggregated to the genus identification. So in this bar, for example, Galway in winter, there's one sample here. All of the ASVs that were in the sample were identified as gyrodinium. So that's what we're looking at. And in general, the Mediterranean sites were much more diverse. They had a greater number of different genera, and they also just had more genera. Both Rio Grande and Galway were pairwise significant with all of the Mediterranean sites. Something interesting we didn't find is we did not find Austriopsis, this red bar in Villefranche. We didn't find it in Rio Grande, and it has been found there in the past in the literature. So this is just sort of a summary of what we just went over. So to discuss key takeaways, there might be some type of riverine influence marine influence for the colonization in Galway. The PLA, it would be interesting to further study the physiochemical properties of that polymer to see if it would be possible to learn more about how it influences biofilm formation. And the majority of toxic PHM and majority of PHM were found in Mediterranean, but it's important to note that there were more samples there, so this could be a sampling bias. There were slight temporal patterns that are interesting to study further. And yes, even though the Mediterranean had the greatest richness, it wasn't overlapping in community composition. There are some study limitations that were really fun and challenging to work with, made it like a puzzle. And just to kind of bring this home, the fundamental question here is one of pollution. They can't have anything to attach to if there's not pollution in the ocean. So with that, I thank you for your time, and my references are here.
Can you take a moment to breathe? You did the 100 meters in the end, but you actually had 20 seconds left, so you could have. Now we can have a discussion, and please talk at a more pedestrian pace. It was very dense, very interesting. I really a lot of work in there, but.
I hope it was going slow enough to be understood.
Absolutely, until the end.
Sorry. Is there any questions at the end? Sorry about
So, Fabio or Lars, who wants to go first?
Okay, I'll go first. Okay, very interesting work, fascinating. Okay, in the sad reality of the huge amount of plastics in the sea. A few things. Okay, first of all, what was the key criterion for the choice of the sampling sites? Was just simply because these were sites where the project partners had the possibility to
Sure, that's a great question. So the project is extremely massive, and these are 5 of the 6 sites which had sequencing performed on samples there, and not all of the samples that were incubated at these sites even were sequenced. So for example, the SEM image I showed you was from Spring and Villefranche. We don't have any sequenced samples from Spring and Villefranche. And then there are other papers that are coming out now from partners with the group about trace metal analysis. Like some of the samples from every site were used for trace metal analysis. So the sample size for sequencing is quite small, and it wasn't my decision to pick these sites. They were predetermined, and my lab was part of the projects that JPI contributed to, and these were the 5 sites where 18S sequencing was successful. There's an additional one with 16S, but they didn't have 18S, and so I just wanted as many samples as possible. I was very sample greedy, and I just wanted to have as much data as I could to try and make comparisons between these different ocean basins, because that's something in particular that for eukaryotes, there's not a lot of between basin comparative studies in the way there is for prokaryotes. So it was the data that was available, but I was really eager to work with that as well.
No, I was asking this because, okay, we know that there are certain other parts of the world that unfortunately host a huge garbage patches. So I think having the data also from other parts of the world would have been nice. But,
well, yeah, I agree. There's many Mediterranean sites and then not so many elsewhere. But when the partners were picking the places to sample, some of them were very industrial, like Toulon was an industrial port location. Naples had a lot of pollution, and Villefranche is it's very secluded, has a very small population, so it was sort of thought as a cleaner site. So the sites were picked for their different features, but some of them are quite close together. You know, it's hard to compare the Atlantic sites because they're in different hemispheres. So it's kind of this Mediterranean cluster, and then one in the North Atlantic and one in the South Atlantic, but I think together still they make an interesting set.
Actually, can you go to the slide where you listed your hypothesis? Okay, no, I see. Okay, the last one is polymer type will influence richness, but not community composition. Is that an expectation that you asked based on previous knowledge?
Based on some studies in the literature, that was our guess, but there are some studies that say it influences richness and community composition. Some say that it influences neither. Some say only community composition. But because the study, the one that I could find that did eukaryotes and PLA, it said that influenced richness and not community composition, I thought that that would be the most likely outcome. And that is what we saw, which is quite interesting. It's still a plastic, you know, it's still fossil fuel based in some degree, but something about the fact that it's partially fermented plant starch, it gives it a different initial colonization, and then when the colonization is more stable at 90 days, it's still more rich than the others, which was very interesting, because people are all about bioplastics, but maybe they could be worse. You know, if PHM prefer them, that might not be a good thing.
And the other thing, look, I wonder if, okay, for this project also, some data from the water column they were taken. I mean, okay, this in order to have an idea if these communities that grow on the plastics, actually, I mean, those the composition reflect the actual composition of this microalgae in the water column, or in a way or another there are some particular species or taxa that attach preferentially
Yes,
to the plastics.
absolutely. So most harmful microalgae are benthic, and so plastic, especially floating microplastic, it offers like a benthic habitat in the pelago. So it's very compelling in this way. And we do actually have water samples alongside from where the plastics were taken. I just didn't have time to analyze them. And there are many, I didn't prioritize it to include in the thesis. Maybe, you know, for the future work, because we're hoping to turn the work into a paper eventually, we could include it, but there are many studies that show that the difference is not significant, that it's very similar, but in the case of benthic species, it might not be the case. So it will be worth looking at. It's a good point.
Thank you very much.
Yeah, thank you.
Congratulations.
Thank you, Fabio.
Yes, thank you for this very nice presentation. It's an impressive project, and it's also very nice work.
Thank you.
So what are your hypotheses for Naples and Toulon standing apart from Villefranche? What would be the mechanistic reason for such a...
Pretty close, and they sandwich it on either side. My best guess right now is that they're more polluted sites. Typically, HAB events happen where there's more pollution. It's kind of a symptom of eutrophication. A lot of HAB events are triggered by large amounts of nutrient input, and Naples certainly has that situation. We do have some environmental data about the sort of setting that the samples were collected in, but I think that to truly understand why Naples and Toulon set apart from Villefranche, we might need to be able to look at a full year in sequencing data from all three sites, which will be, maybe it will just take longer to get that. You know, I'm sure that it's not the last sequencing type of project that will be happening in the area. So my current idea is that it might be due to the fact that there's higher nutrients in these regions, and through a very simple PCA on the environmental variables that I didn't have time to present all the results from my thesis, but I have a couple, maybe I'm opening a can of worms by bringing these in, but in in Naples, which had the most samples, it had the full year, every single season had many robust samples. The most important variables were actually mostly nutrients, which was not what we were seeing in the other sites. It was usually temperature and salinity because they were maybe discontinuous seasons. And so that really big, strong signal of temperature was coming through first. But in Naples, we get the more subtle signals from nutrients in the first in the significant dimensions, which were the first four for Naples. So I think that the nutrient story is very important when we're considering the toxic and harmful microalgae within plastics.
Thank you. Then my second question is about, I did not see the effect of the incubation time. What is?
Oh, sure.
Did you see any succession of population or?
In Naples in particular, there was a really interesting signal. I will say it again a little bit slower, and I didn't get to mention that it's happening in winter as well. So we have the spread of, the distance between these samples. They're most similar when they're coming from, the same time period in autumn. So we've got the 7-day over here, we have the 30-day kind of moving more gradually into this cluster of 90-day samples. So there is a sort of seasonal gradient in the differences between these samples. And we see it similarly in winter, where we've got the end of autumn up here. We've kind of got a mix of 30 and 7-day samples kind of stretched apart from the 90-day samples of winter. So there is this sort of, I mean, visually at least, there is this signal, and the incubation time given season in Naples had a contribution of about 13% of the variance and had a significant p-value, but we didn't have enough samples of each time period in every season to get a really strong answer about this. We would have actually, I calculated, we would have needed 900 samples to be able to statistically test every single layer of our variables.
So if you understand what here, either that you start incubation what are the triangles?
So 7, 30, and 90.
No, the color are the seasons.
So spring is light blue. So spring is actually quite hard to see because it's completely inside winter. Winter is here, this is summer, and then this is autumn.
Okay, so whatever is the season that autumn and winter, you start from far from the final community composition.
Yes.
And you converge to one community composition, which is a kind of
like more steady for the
steady and alike between seasons.
Bless you. I see a sort of interesting story, especially in the Naples data, because winter is a season where the temperature is getting very cold, and so we've got the sort of, it's not as stratified in the water. We have the nutrients coming up, but it doesn't really have the temperature to support a bloom. And then spring is kind of winter 2.0 in a way. It's still quite cold. It's warming up a little bit, but it's not really until the end where we're reaching that spike in temperature that comes in summer, which is sometimes too hot. Stratification comes, but then in autumn, we've really got that sweet spot of the temperature declining a little bit, still warm, and more nutrient kind of availability. But in the situation where we've got, the end of autumn, the start of winter, and then the end of winter relating to the start of spring, it seems like there is sort of some type of, maybe, I don't want to speculate too far, but maybe some type of seasonal sort of cyclic relationship with the way that the seasons and those environmental variables that they stand in for really are related to the community composition, and particularly this group of organisms. It would be really interesting to look into it further. And I'm all ears.
You are motivated. Thank you.
Thank you.
Okay, the samples of the different seasons were collected in different sampling dates or all in the same day?
So it was like a synchronized sampling effort for an entire year between all 5 sites, and of course,
no, but okay, for example, in Naples, the samples of each season were collected in just one single day? Or?
Yes, all of the times 7 samples come from the 7-day into autumn. Yes, exactly.
Then I wonder if that thing, probably such a big variation for autumn, might reflect really some kind of spatial variation rather than really the seasonal.
They are, I think they're from 1 or 2 cages that are in the same location. So I do think that over here it's quite interesting that this is kind of right on the tail end of summer. It's a very unique group, and it's so strongly clustered together. It's very interesting.
Right. Thank you very much. So can I ask you to sit for a moment? Well done, everyone who's presented today.
Yes.
Be kind to your colleagues that are presenting tomorrow, especially tonight at the dorms. Yeah, go far away if you need to. I just want to ask you all, if we could all move down outside, just where we had lunch. We're going to take a group picture with everyone. So before you go to the bus, stop there first. And we should go there now and meet all the other groups. Thank you very much.